Welcome to the lecture on neuro, muscular, eye, and ear. So I want to give you a little bit of a in-depth dive into what's happening on the neuromuscular and what's happening with the eyes and ears. So the ability to feel pain, no matter how unpleasant, it makes us actually aware of the world around us and can prevent much greater damage in the long run. Here we have an individual, Gabby Gringas, shown here at 3 years old. She was actually born without the ability to feel pain. And this condition actually puts her at greater risk for injury. So as much as we don't like pain, it actually is necessary. So let's look at some of the functions of the nervous system. The nervous system actually is present in almost all multicellular animals, and it has three primary functions. Number one, to receive stimuli. The nervous system collects information about the internal and external environments, and then it processes that information. And for us humans, we process that information in our brain. So we interpret that incoming stimuli, and then we determine a response. And from there, we actually initiate the response. So the nervous system sends signals to muscles and glands in response to the internal and external environment. But I want to take a closer look at the structure of a neuron. So neurons are actually individual cells that specialize in carrying electrical signals, and they're the building blocks of the nervous system. They have three very distinct elements. Number one, the dendrites. And the dendrites actually sense and respond to stimulation from outside the cell and send that information towards the cell body. And then we have the cell body, which contains the nucleus and other cellular machinery, which you already are very much aware of what's inside the cell. And so that signal continues down through the axon. And the axon is this long tube-like projection that extends from the cell body, and it transmits the signal to other cells. Then we have these support cells, which are known as glial cells, and they're a whole family of cells that actually support the neuron itself. And both they protect, insulate, and nourish the neuron. So they have different functions, and we've given them different names, but they all come under the category of glial cells. So there's different types of neurons. There's sensory neurons. And sensory neurons collect information from the animal's environment. That includes us, right? And dendrites are modified to respond to external stimulation such as temperature, touch, taste, smell, light, or sound. These are all perceived by sensory neurons. Then we have motor neurons. And motor neurons actually initiate the response to stimuli. They stimulate action by conveying signals to muscles or to glands. Those are motor neurons. And then we have what's called interneurons. And interneurons interpret signals coming in from sensory neurons, and they actually relay them to motor neurons. So they're in between the two. That's why they're called interneurons. Interneurons are located only in the brain and the spinal cord. But let's take a more sort of global perspective of the nervous system. So we have the central nervous system, which is composed of interneurons and other supporting cells that process the information, and then they send it to other cells, right, to determine what to do. So our central nervous system is comprised of the brain and the spinal cord. Then we have what's called the peripheral nervous system, and this detects stimuli and transmits signals. And I'm going to see if I can get my mouse to kind of show you where I am. The peripheral nervous system is right outside of the spinal cord. So it's pretty much the rest of the nervous system that isn't the brain or the spinal cord. But we have different categories of the peripheral nervous system. So when we talk about sensory pathways, these are the sensory neurons that we talked about already that receive information from the environment. Then we have our motor pathways. These are the motor neurons that transmit signals to the muscles and the glands. And within that category, we have the what's called somatic nervous system. These are the voluntary motor pathways. So this is just terminology that I'm explaining to you. So the somatic nervous system relays signals to skeletal muscles. And the somatic nervous system, the voluntary, which means we can control it, it's under conscious control. I can move my arm, I can move my leg, I can tell it when to move. But then we have the autonomic nervous system, and this is the involuntary motor pathways. And so these pathways relay signals to glands and to smooth muscle tissue and to cardiac muscle. These are not under conscious control. Remember, I don't have to think and remember to tell my heart to beat. I don't have to think and remember to tell my glands to secrete different hormones. It's not under conscious control. Now, under the autonomic nervous system, we have two big categories. Again, terminology. We have the sympathetic nervous system, and this is responsible for coordinating the body's fight or flight response to stress. That is the sympathetic nervous system. And then we have the parasympathetic nervous system, and this is responsible for controlling activities related to digesting food and eliminating waste. So let's take a look at the overview of the nervous system and their interaction. So here we're going to take a look at what happens when we're, for example, bitten by a mosquito. So first we have the stimulus, which is the bite of the mosquito. And then a sensory neuron picks up that sensation on, say, your hand as an irritation. And it sends a signal to convey this information to your spinal cord. So here we have the stimulus, and the sensory neuron is picking up that stimulus. It sends it to an interneuron, which sends the stimulus to the brain. And then your brain interprets that signal as, hey, that is a mosquito biting me. And then it passes the information to swat the mosquito, the response, to another interneuron, which interacts with the spinal cord to actually communicate with a motor neuron. And the signal is sent through the motor neuron to a muscle, causing you to swat at the mosquito. So you can see and follow the pathway from stimulus to cellular response. But you'll hear something called a reflex, So reflexes can generate a response to a stimulus without need for processing the signal by the brain. And that's because, as you saw me walk through that pathway, what you probably realize is there's some lag in there, because we have to go from the stimulus to the sensory neuron, to the interneuron, to the brain, again to an interneuron, to a motor neuron, to a response. There's lag. And if there's something that is imminent danger, you may not have time for that lag before you actually incur damage. And so in order to compensate for that, we have something called reflexes. And so reflexes can actually generate a response to a stimulus without need for processing the signal by the brain. We don't have to have that lag with the brain interpreting what's going on. The hand just will reflex move, or the leg will reflex move. So if, for example, you put your hand in fire, you need to immediately withdraw that hand, and that's what the body does, and it doesn't wait for the lag of the brain to actually interpret it. And so reflexes are very important because they enable an organism to respond faster to an imminent danger. You can process what happened later. But let's take a closer look at what's happening when a dendrite of a neuron receives external stimuli. So here in the first image, we see the cell membrane. This is the neuron. We see the stimulus. We have these positively charged ions that are outside of the cell membrane. And we have the receptor proteins and these ion channels. So step one, the receptor proteins within the cell membrane of the dendrite respond to stimuli by briefly opening up the little channels. So the channels open. Now, the open channels allow the charged ions to enter the dendrite, momentarily altering the electrical charge within the cell from the negative charge that it carried prior to the stimulation, because all of these positives are going to rush in. And then signals from all the dendrites of a neuron converge. So it's not just one neuron that this is happening to, it's all the dendrites that are actually interpreting this on this cell body. As you can see when you look at the image of the cell, it has more than one dendrite. And so the signals from all the dendrites of a neuron converge, and if the sum total of these signals coming from inside is significantly positive, then the cell initiates an action potential that travels down the axon. And so this is really important, and it's actually kind of a cool feature, because if you have just one little dendrite that basically is signaling that it has a positive charge, well, that's not really enough for the cell to initiate an action, because that might be an error, that might be some sort of false signal, right? But if all the dendrites of this cell, or some total of them, begin to signal this positive charge, then that most likely is real. And so that causes the cell to initiate an action potential, which travels down the axon. Okay? And so let's look a little bit closely here at another term that you'll hear called the resting potential. I'm at the top right here. So these positively charged ions tell the neuron fire, right? Tell the neuron downstream that something has happened, right? And while the negatively charged ions tell the neuron don't fire. So positively charged ions, fire. Negatively, if you're mostly negative, don't fire. And so we have this resting potential, which is prior to stimulation, there's a greater positive charge outside the cell than inside the cell. This difference in charge, called the resting potential, is produced by active transport of ions and results in a polarized cell that's ready to fire. So that means that now mostly positive ions are outside the cell. The cell is ready to now fire again. If the stimulus comes through, which causes these positive ions to rush in, then we determine whether or not we've got enough dendrites that are converging, and if the sum total of these signals are actually significantly positive, then we fire, and then the action potential travels down the neuron. So let's take a look. Again, we're going to go out just a bit from that close look at what's happening to actually cause a neuron to fire. Let's look at the outside of the neuron. Here we've got the neuron, and I told you there is an axon, as you can see here. But these axons actually, some of them have a sheath called a myelin sheath. And the myelin sheath prevents the action potential from weakening as it travels down the axon. And so this is electricity, right? This is basically a current that is moving down the axon. And just as current that we have in walls with the electricity, we put insulation onto those wires so that we can keep that action potential, that level of current, relatively the same level as it's going down the length. Because normally, as electricity travels, the longer it has to travel, the lower the actual current state becomes. So you may start at, let's just for example say a 5 at the start of a level 5 of electricity, but if you have this very long span that the electricity has to travel, by the time it gets to its endpoint, it may be a 2. And to prevent that, we insulate both our wires that we humans use, or in nature, it's insulated with a myelin sheath in order to prevent the action potential from weakening as it travels down the axon. But it turns out these myelin sheaths, which are basically a fatty coating that surrounds the axons, they have the appearance of being white. They look white. So areas of the brain that are rich in axons that appear white. And then we have regions of the brain in which cell bodies are, and the dendrites, they appear gray because they don't have any myelin on them. And that's where you get the terminology of the white matter of the brain and the gray matter of the brain. So I'm showing you an image here, which is a real life slice of brain. And you can see the appearance of white and gray matter, stained white and blue here, respectively, in this section through a human brain. And it reflects whether the tissue consists mostly of axons, which is the white matter because it has the myelin sheaths, or mostly cell bodies and dendrites, which are called gray matter because they don't have any myelin sheaths. So that's where you get the terminology, the white matter of the brain and the gray matter of the brain. But I want you to take a closer look at what's happening at the synapse, because this is really important to understand as you try to interpret what's happening with a drug. We have reuptake inhibitors, etc. Well, what does that mean? So let's look at what's happening at the synapse. So here in step 1, what we're looking at is when the action potential reaches the axon terminal, then the neurotransmitter filled vesicles that we have here that we're seeing at the top, these vesicles are filled and they fuse with the presynaptic membrane, releasing these neurotransmitter molecules into the synaptic cleft. And the synaptic cleft is this space between one cell's axon, one neuron's axon, and another neuron's dendrite. So we're passing information. As you can see here, if you look at these two cells, what we're doing is just sort of zeroing in on this communication in the synaptic cleft between these two neurons. So one cell at the top, the blue cell, has received a stimulus, and it was enough to actually generate an action potential, and that action potential stimulated these vesicles filled with neurotransmitters to fuse with the blue neurons membrane, presynaptic membrane, and release the neurotransmitter molecules into the synaptic cleft, as we see here in this step 2. So we can now see the neurotransmitter molecules that are in the presynaptic cleft. And these neurotransmitter molecules, they diffuse away from the axon and they bind to the receptor sites on the postsynaptic membrane of the receiving cell, the receiving neuron, as we can see here. And then when a neurotransmitter actually binds to the receptor of this receiving neuron, it causes a channel to open, allowing the ions to flow into the adjacent cell, either stimulating or inhibiting it, depending on what the neurotransmitter is actually for. And so we've got these positively charged ions that are also in that cleft. Now, so when that happens, as we know from before, when that actually stimulates that yellow neuron to perform an action based on what the neurotransmitter is telling it to do. But the neurotransmitter can't stay there forever, otherwise the cell would never stop that action. It would continue to perform that action, right? So somehow we have to think about, well, what are we going to do with these neurotransmitters that are now in the presynaptic cleft in order to basically have some control over the situation. So the neurotransmitter molecules are then, released from these postsynaptic receptors, and then they're either taken back into the blue neuron, the releasing neuron, so they can be reuptake, right? We could have reuptake happening, or they can be recycled by the initial axon terminal from which they were released, or they can be broken down by enzymes and completely cleared from the synapse so that the synapse can be used again. So they just don't stay there in perpetuity. They can, the transmitting neuron can reuptake those neurotransmitters so that it can, wait for another action potential to release them again. Or we can have enzymes that come into that presynaptic cleft and remove the neurotransmitters in their totality so that we're ready to receive another signal. Okay, so at synapses, not all signals are propagated. Some sensory noise may actually be filtered out, and this helps to reduce the barrage of stimuli to the brain. And if you look at this image, what it's showing you is, you look at all these people. Here we've got the basketball player. He's got to focus on getting that ball into the net, and we've got all these people behind the actual basketball net waving these balloons and just trying to cause a lot of distraction, a lot of noise for his brain to try to filter out, hopefully causing him to make an error, right? But there's a special neurotransmitter called acetylcholine. And by preventing acetylcholine release, this is how the botulinum toxin actually works. Botox. It's actually a toxin from botulism. And botulism basically paralyzes muscle. So if, sort of in the wild, you were to, say, open up a jar where the botulism organism had actually began to grow and make its toxin, and you ate that food, you would suffer a flaccid paralysis. None of your muscles would work. And that's because botulism toxin is preventing acetylcholine from being released, telling the muscles to work. We use our knowledge of what's happening. We do this all the time. When we learn something in science, we try to use that to make life better. And there is an organism called botulism, and it creates a toxin. Normally you'll find botulism growing in jars of food that have been improperly processed. And so it starts to grow. But what this botulism toxin does is it actually will cause what's called a flaccid paralysis of the muscles, which means the muscles cannot move. And the reason it causes this flaccid paralysis is because it's preventing the neurotransmitter acetylcholine from being released. And if that neurotransmitter is not released, of course, as we know, then we can't get the firing. We can't get the action potential to happen to cause the muscles to actually contract, right? And that's bad. That could kill you because, as you saw in our respiratory lecture, we use the muscles of our diaphragm and the intercostal muscles, the muscles between our ribs, to breathe, to bring in air, right? We also use the muscles of our heart to pump our heart. And so if suddenly we cannot release any acetylcholine to talk to the muscles to tell them to contract, then you're going to die because your heart's not going to beat and you are not going to be able to breathe. So this toxin is very powerful in large amounts. However, that flaccid paralysis, someone not too long ago actually discovered that we could inject a small amount of this botulism toxin into the muscles of, say, the forehead so that they cannot raise and cause wrinkles, and suddenly Botox is the world's high-performing reducer of aging and wrinkles. But it does have some cautions with it as to amounts and actually, where you can place it in the body, where you can inject it. All right. So let's talk about the five senses. So we've got hearing, touch, taste, smell, and sight. So our senses, hearing, touch, taste, smell, and sight, are windows to the world. That's how we actually perceive the world outside our brain. And each of these senses brings us information about a different slice of the world. But the senses have a lot in common. So we can have these variations in sensation intensity, meaning that I can tell whether I'm holding a cotton ball or a bowling ball. And that's because if I have this, for example, lightweight ball, I'm actually going to get this weak stimulus. it's interpreted as a less intense sensation by the brain. And if I'm holding a bowling ball, as you can see by the number of neurons, sensory neurons that are actually being stimulated, then I interpret that as a more intense sensation. So we can experience these gradations of sensation depending on the number of neurons that actually fire. And so it allows us to make determinations of the things that we're actually touching and handling. But let's take a look at how smell happens. How do we smell? So this is a look at the nasal cavity of a human, and we're taking a sort of a blown-in picture, a close-up of what's happening, and we've got these chemoreceptor cells that are sensing the different smells as chemicals, and because they're chemoreceptors, chemical receptors, they're able to take that as a stimuli and move that stimulus down through the neurons as we saw before, and it's sent as an action potential to the brain, and we can interpret what we're smelling. Well, it turns out that dogs have as many as 40 times more smell receptors, or these chemoreceptors, than a human. Hence their keen sense of smell, which gives them tremendous proficiency at detecting things that we have a very difficult time detecting at low levels. So they can detect drugs, in luggage, explosives on a car, where we would not be able to detect them at all. But let's take a look at the eye and how our eye works. So here we have the globe of the eye. We are looking at one slice of it. To the far left, you've got this clear area, which is called the cornea. Light comes into the eye through the cornea. We've got a region here called the iris. This is what imparts color to the eye. And the opening between the iris, which is like a circle if you were to be looking straight on at the eye, and we call that the pupil. And so the iris basically can open like a lens of a camera. It can open up to let more light in, or it can close down to a pinpoint to let less light in. So the iris is basically controlling the amount of light that's coming into the eye, and also it's imparting the color that we see to the eye: brown eyes, blue eyes, etc. Right? But that light, as it comes through the pupil, through the cornea, through the pupil, it hits the lens. Now the lens, the job of the lens is to focus that light on the back of the eye, where we have these special receptors that help to interpret that light. And so we call this region of the eye that is actually interpreting the light the retina. And the retina is composed of rods, and these are special photoreceptor cells that are highly sensitive to light, and they allow for vision at night and in low light situations. And then we have cones, and these are special photoreceptor cells that are sensitive to color. And so they can be either red, green, or blue sensitive. And so malfunctioning of the cones is actually a genetic condition that is more common in men. Usually it's carried on the Y chromosome, so it is sex linked. And it's more common in men than women, and this is what leads to color blindness. Okay. And so let's just take a look at how these photoreceptor cells actually are working. So the light is coming in. We're just looking at one photoreceptor cell here that we've pulled from the retina, right? The light's coming in. It's come through the lens of the eye. It's hitting this photoreceptor cell. We've got this stimulus that's happening, basically the same thing that's happening before. We've got a sodium channel here. And so light energy causes light-sensitive molecules to change shape, closing the sodium channels in the photoreceptor cell membranes. And the closing of the sodium channels reduces the amount of inhibitory neurotransmitter released by the photoreceptor cells. So now, when no longer inhibited, the adjacent neurons excite the neurons that signal the brain. Okay? And then an action potential is sent through the optic nerve, and light is perceived by the brain. So that's basically what's happening inside the photoreceptor cells to actually trigger the action potential to which the brain then perceives and interprets. But not all organisms have the same eye conformation and organization that we do. So here we have, we're going to take a look at a few different types of eyes. Here we have a flatworm, and they have these eye cups. And their eye cups contain these photoreceptor cells that are able to detect the presence and intensity of light but cannot form images. And that's a flatworm and its eye. When we look at a fruit fly, for example, the workhorse of the science lab, they have these compound eyes. And so the fruit fly, their eyes actually contain many separate light sensing units, each with its own lens. Remember, we only have one lens in the eye. Each of their units actually has a lens that directs light onto about a dozen photoreceptor cells. So they're sensing a much different world than we see. But let's look at the eye of a squid. And this is really interesting because their eye is has the same conformation anatomy as ours. It's a single lens eye, and that's what we have. And so it focuses light onto these highly sensitive photoreceptor cells back at the back of the eye. So they're viewing the world very much as we do. But when we look at insect vision, this is really interesting because how we see it, for example, on the left, if we were to look at this silverweed flower in daylight, it looks yellow to us. But an insect actually is able to see UV light, ultraviolet light. And so it looks very different to them. And here we can see this flower that just looks yellow to us, has this spectrum of white and red and blues that we cannot see. So some flowers actually build ultraviolet light reflecting petals that causes insects, which can see this part of the spectrum, to come to the nectar, ensuring pollination of the flowers. So they're not actually trying to attract us. So they're trying to attract the insects, and so there'll be this completely different spectrum that they'll actually use in their petals that are sensitive to UV light. So they're signaling specifically to the insects. But here's what's interesting with insect vision. If you've ever walked down a beautiful forest path, or even walked down the path, in your yard, and here you've got this spider who has spun a web across the path, and you walk into the web and you think, oh gosh, that's gross. And why would a spider, spin a web across the path? And it's super interesting because they're trying to catch insects, right? And they will spin this cross-like pattern. Here we're looking at the St. Andrew's cross spider, and they will actually spin using reflective silk that reflects in these certain wavelengths of light, causing this UV reflection that looks like petals of some flowers in order to lure the insects. So if you were to look at this spider and you actually kind of look at it from afar, and you could see the UV pattern that he's woven into the web and along with its body kind of hanging in there, it actually does look like a flower petal. And so moths and butterflies would be drawn to it and think that they're coming to a flower instead of coming into a spider's web. So let's take a look at how we hear, how that actually occurs. So number one, here we have the entrance to the ear, and we have sound waves that are being funneled into the ear canal. And so the outside of our ear is very important to help us hear. That's capturing sound. If you wanted to hear a little bit better, you would sort of cup your hand around your ear to capture more of the sound waves. So the sound waves are funneled down the ear canal. Then the sound waves cause the eardrum, which is here, to vibrate. And in step 3, the vibrations pass to the inner ear membrane. And then we have this fluid in the inner ear that's actually conducting the vibrations. And it's bending these hair-shaped receptors and releasing the neurotransmitters. So it's causing these receptors that we can see here at this blow up in step 4 to actually receive the signal because of the vibration that's coming from the fluid in the middle ear. And then these neurotransmitters that are released cause the auditory neurons to fire an action potential. And then that signal is passed to the brain where it's interpreted as sound. So we can see it's actually a very interesting anatomy to the ear itself. And so just going to go through some terminology so you can see what you're looking at. Here we've got the sound waves that are coming in. Here we've got the eardrum. We've got these little bones that are inside the ear, and these are some of the smallest bones in the body. Here we have the inner ear, which is comprised of the cochlea and the semicircular canals. And then you can see in the blowout of the cochlea, that's where we have these fluid-filled chambers where we're actually causing the vibration, which causes the stimuli, which basically is starting that action potential, which then goes down from the auditory neurons to the brain to actually signal an interpretation of sound. However, chronic overstimulation due to loud noises can damage the hair cells in the inner ear, because we just saw how important those hair cells are. And they can damage the hair cells in the inner ear, reducing their ability to stimulate auditory neurons, and this is a leading cause of hearing loss. And this begins to happen at about 80 decibels. And so you never want to be in an environment that has 80 decibels or more because you are incurring hearing damage at that point. And we see that quite a bit with individuals who are members of a band, and so they're always exposed to this greater than 80 decibel sound. I don't know if you've ever gone to a live concert. If you are fairly close to the stage, when you come out of that concert, it feels like you have cotton or water stuffed in your ears for about a day or so, and that's because you actually are causing some damage to those hair cells. And if you completely break them off, you may not get them back, which means that that damage could be permanent. So you always want to protect your hearing, and we see that more and more with musicians now. They'll actually have hearing protection in their ears to prevent them from going deaf. But there's different ways to perceive the world other than the way that we perceive the world. There are some animals, organisms that have this ability to do echolocation. So bats are able to sense where things are in their environment, even in the dark, by emitting these high-pitched squeaks and then waiting to hear the sound as it bounces off the surface around them. And so this echo response, they're able to actually locate and determine the size and the shape and the distance of organisms in their environment. But let's talk a little bit about touch. So what's happening with touch? So here in this example, we've got, say, a thumbtack that's actually pricking your finger. And so what we're going to sense at that point is the mechanoreceptors, which are sensitive to pressure, are going to initially feel the pressure that begins to impart on the tip of the finger. And as it goes deeper, we're getting more and more stimulation. If it should have some temperature to it, there's also thermoreceptors that we have that are sensitive to temperature. And then, of course, we've got the pain receptor, which is sensitive to pain. And so we're getting, essentially, we could have all three of these firing down and the line to cause an action potential to the brain, saying, ouch, I've got some pressure going on, to it's a, say, ice cold or red hot, piece of metal, and then the pain receptor fires and we interpret it as pain. And of course, then after the interpretation, we have the action that's sent back from the brain to withdraw the hand to the motor neurons. So here we're just taking a closer look at what's happening in that cleft. So when we have touch, we can see that we've got our positively charged ions that are there in that synaptic cleft. And so this external stimulation, such as the prick of a pin, causes this change in the shape of the neuron's membrane, altering the permeability to charged molecules and initiating the action potential, right? And we're just here looking at the pain receptor proteins at this point that are here on the outside as well. So it's a very sophisticated system. But what's interesting is, the way other organisms on the planet actually can perceive the world. We talked about echolocation. And a lot of times, just, as a point of interest, people who have lost one sense, for example, the ability to see, can begin to increase their use of their other senses to help them perceive the world. And you'll see individuals who have, a cane, if they are blind, and you'll see them tapping with the cane, and they are also trying to listen for the bounce of sound. I've heard individuals who are blind be able to interpret what environment they're in based on how the sound is bouncing off walls or bouncing off the ceiling or bouncing off the table. They can determine if there's a wall near them, if there's a table in front of them, not only with their touch, but basically how the sound is responding to them. But let's take a look at some other ways to evaluate the environment that an animal can use. So some animals actually, especially migratory species, here we're looking at a beluga whale, they're able to detect the Earth's magnetic field and use it to help them navigate. Here we have a snake, and they have special openings on either side of the head, and it opens into what's called the pit organ. And so these pit organs, actually, they're located on either side of the head. And here we're looking at a pit viper. And these pit organs allow them to sense a prey animal even in complete darkness, because what they're sensing is the body heat that is coming off of the animal which they are hunting. And here we've got a cute little penguin surfing. Let's take a look at the skeletal muscles and how the muscles work. We talked about the nerves and the action potentials and how that happens. And eventually we're innervating the muscle to cause a response, but let's look at muscles, how they work. So here we're looking at the skeletal muscle and how it generates movement. So we've got the point of origin. Here we're looking at the scapula and the radius. That's the shoulder blade and the upper arm. I'm sorry, the scapula and the humerus. My mistake. And so we've got muscles which are attaching to bone by tendons. Tendons are making that attachment. Here at the top, we're looking at the bicep muscle that everyone's very familiar with, the back of the arm. We have the tricep muscle. Here, the upper arm bone that they have pointed out, which is the humerus. And then we have these insertion points of the tendons. And so you always need two muscles working in opposition so that the arm can basically contract towards the body, right, and lift something up. Let's say you have a dumbbell in your hand and you want to actually lift that weight, then you're going to use your bicep to go ahead and contract and lift it up. But in order to pull your arm down, if you didn't have a tricep muscle on the back of that arm, you would be stuck in that flexed position, right? So we need the tricep muscle that's on the back of the arm in order to flex and in order to contract, and then we extend the arm. So muscles working in opposition of each other give us the capability of flexion and extension of our bones. So we can move the arms, we can move the legs. But let's take a closer look at what a muscle is. When we say a muscle, we have a concept of what it is, But let's take a closer look at what actually composes a muscle. So here we're taking a closer look at the biceps muscle, and we can see as we kind of go and take a look closer and closer and closer, the bicep muscle is made of these bundle of muscle fibers. And then we have this muscle fiber gets smaller and smaller, and we can begin to see the nuclei of the muscle cells. And then we get down to what's called a myofibril, myo being muscle, myofibril. And then we take a closer look at what's happening to actually cause the contraction, and that is the contraction's happening in what's called the sarcomeres. And these sarcomeres are made of actin filaments, which you can see pointed out here, and myosin filaments. And together the actin filaments and the myosin filaments compose the sarcomere. And there may be 100,000 sarcomeres in one biceps muscle. And so let's look at how we actually get contraction. So a sarcomere shortens in a 4-step process. Contraction is because these sarcomeres are shortening, right? So number 1, detach. So a link between a myosin and a parallel actin filament is broken as a molecule of ATP binds to the myosin, as you can see here. And I won't need you to remember these steps. Just kind of get a basic understanding of what's happening. If you go further in anatomy and biology, of course you will have to memorize what's happening at the sarcomere level. But for now, I just want you to sort of take it in and have a basic understanding of what's happening, that this contraction is happening at the sarcomere level. So now we have this molecule of ATP that has bound to the myosin, and then this link is broken, and as the ATP breaks down, energy release alters the shape of the myosin into a higher energy shape in step 2. And then in step 3 we get reattachment. So in its altered shape, the myosin reaches further down the actin filament where it reattaches. And then in step 4, the myosin then snaps back to its original shape, pulling the actin filament as it does so and shortening the fiber. So here we've got the relaxed sarcomere, and we can see what is happening as we get the contracted sarcomere. Okay. So let's take a look at the functions of the skeletal system. Right, it's not just to move around or lift up weights. So number 1, we've got structural support. So it's providing shape and structure, including securing organs in place. So it's actually giving us protection of vital organs such as our heart and our lungs, our brain. And then it also is enabling movement, as you know. But the skeletal system is a major part of our red and white blood cell production. And that's happening in the bone marrow, as we saw before when we talked about blood. But it also serves as this mineral reservoir. And so elements such as calcium can be released from or stored in bones in response to deficiencies or excesses in the bloodstream. So if you get too much calcium in the bloodstream, too much calcium can actually stop your heart. Your body's going to try to prevent that. It's going to start to sequester some of that calcium in the extra calcium in your bones. And that way, if you come to a point or a time in your life where you are deficient of calcium in your diet, you're not taking in enough. It rarely happens in America, but in other places of the world, this is certainly a possibility. And so the body will actually reach into these calcium stores that are both in the bone and also the teeth and begin to use the calcium that's stored in the bone. And the first thing that you'll start to see is people will begin to lose their teeth because your tooth structure is held in to the bones of your jaw. You also will be subject to softening of the bones, which means they'll start to bend and break because the body is using that calcium to function, because calcium is part of what we use to actually cause the heart and other muscles to contract. And so storage of mineral reservoirs is also another important component of the skeletal system. But again, not all organisms have skeletal systems in the way we have a skeletal system. So there's something called a hydrostatic skeleton. So this is a fluid-filled cavity with sufficient rigidity so that muscles surrounding it can generate movement. An example of a hydrostatic skeleton is this jellyfish. We have exoskeletons in the animal world. Here we're looking at a lobster. Crabs also have it. You're very familiar with these. Ants, spiders, exoskeletons. This is a rigid outer covering that supports and protects an animal's body. And then we have the endoskeleton, which is what we have, right? It's an internal support structure composed of hard mineralized tissue, and it's inside the body, not outside the body, as with an exoskeleton. But what is osteoporosis? You've heard of people, especially women, as they get older, they develop osteoporosis. Well, osteoporosis actually results in reduced bone density and a change in the chemical composition of bone, leading to an increased risk of bone fracture. So the density of the bone begins to decrease, go down, and that leaves an individual who's experiencing osteoporosis, osteo meaning bone, to be at risk for fractures. And the fracture, as you know, that we see happening most commonly in individuals with osteoporosis is a fracture of the hip. All right, so we're going to start to piece some of the pieces of all this puzzle, bits and pieces together. So that's where you should be right now in your learning, where you're starting to fit all that you're learning together to have a greater understanding of what's happening in the biological world, especially with physiology. I do want to take a closer look at the brain. And I want to start with, even though I'm going to give you, quite a bit of information on the brain, I'm staying very broad for this fundamentals course. There's a lot more that we know about the brain, but for as much as we know about the brain, there is infinitely even more that we don't know about the brain. All right. So the brain has, we have terminology that goes along with it. And here, let's go through our terminology. So basically, it's broken into three parts. We've got the forebrain, which is composed of the cerebral cortex, the thalamus, and the hypothalamus. And that's shown in blue. And then we have what's called the midbrain, that's shown here in purple. And then we have what's called the hindbrain, that's shown in this green. The hindbrain is composed of the cerebellum, the pons, and the medulla. So now let's take a closer look at what each of these parts actually, their function. So starting with the forebrain, the cerebral cortex is involved in abstract thought, problem solving, and language. Also in the forebrain, we have the thalamus, and the thalamus receives sensory input and relays some signals to the cerebral cortex while blocking some others, so it's kind of working as a gatekeeper. And then we have the hypothalamus, and this is an extremely important portion of the brain. It regulates many fundamental drives to stay alive, including hunger, thirst, sexual activity, maintenance of body temperature, controls of hormone secretions in the tiny pituitary gland is controlled by the hypothalamus. So that is the forebrain. Now the midbrain, what we see here in this purpley color, filters and evaluates motor and sensory neuron signals. So I'm going to use sort of the pointer here, possibly you can see it. And then let's talk about the hindbrain, the cerebellum. This is where we coordinate our motor activity. In the pons, this is the pathway for motor and sensory neuron signals. And then we have the medulla. And this is the pathway for motor and sensory neuron signals as well, but it regulates basic physiological functions such as respiration, heart rate, digestion, which means if you have damage to your medulla, your heart may stop because it's regulating your heart rate, or your heart rate may speed up dramatically because it's regulating your heart rate. If you have damage to your medulla, your respiration can actually change, right? And we actually look at some of these cardiac, heart, and respiratory changes to tell us whether or not we have some damage to the brain, because we could see different patterns in breathing that are what's called pathognomonic, which means a very specific indicator for a type of damage to the brain. So after complex muscle movements are learned, such as the precise sequence of knitting, they can then be done without much conscious direction on sort of this autopilot as the cerebellum directs them. So this is a learned motor function. And this is why athletes train, right? So that they can actually have this learned motor response where the cerebellum will actually just do what you have trained to do, and you can use that cerebral processing to do something else, right? But the brain also has what's called these pleasure centers. And so quite some time ago, many years ago, a researcher by the name of James Old set up this apparatus that allowed an animal to administer stimulations to its own brain by simply pushing a lever, which means that these rats actually became lever addicts. If you were to put that stimulation in the pleasure center of the brain, they would actually push that lever more than 100 times a minute for hours on end. But I want to take a look at more terminology of the brain, and this is looking at regions of the cerebral cortex in particular. So here we've got the frontal lobe. Now this is really important because the frontal lobe regulates speech production, motor control, smell, problem solving, and many aspects of personality are here in the frontal lobe. And it turns out the frontal lobe can actually be accessed through the nose. You would go up the nose and break into the bone, and you have access to the frontal lobe. And this is how people many years ago would do this horribly crude, it would never be done today, thing called a frontal lobotomy, where they would just literally take a wire, go through the nose of an individual, scramble around their frontal lobe if they were having these psychological events, and basically completely wipe out their personality, and they would just walk around with just being, without any personality whatsoever. And that was called a frontal lobotomy. We do not do that today. It's very, very wrong. But that's the region of the brain that they were actually basically destroying. And so there's another region of the brain called the temporal lobe that we see here with this pointer. And your temporal lobe is, running alongside where your ear is. And in your temporal lobe, you perceive and process auditory, that means sound, and visual sensations. And it's important in pattern recognition and language comprehension. And so if someone hits you very hard on the side of the head, they can actually damage, you could lose sight because that's where you're processing and perceiving some of your visual sensations. You could, of course, lose your hearing, not only by damage to the ear and the eardrum itself, but by damaging the brain if the concussion, if the blow is hard enough that it's damaging the temporal lobe. And so you can lose vision through damage of your temporal lobe. Then we have what's called the parietal lobe, as we can see here with the pointer. And the parietal lobe receives and perceives touch and pressure sensations, and it's important in sensory integration. So it's sort of managing all of that sensory information. And then we have the occipital lobe, which is at the back of the head, and it's, again, receiving and processing visual information. And the occipital lobe is where you're doing the great majority of your visual processing. So again, a blow to the back of the head could cause blindness, right? But where are the speech centers in the brain? What part of the brain am I activating right now in order to talk to you? So we're looking here at what's called Broca's area, and this region coordinates the muscles necessary for speech production. And then we have Wernicke's area, which is responsible for language comprehension. So when someone is talking to me, I'm comprehending that language using Wernicke's area, but also I have to comprehend what I'm saying in order to speak to you. So two regions of the brain have to work in concert for speech. Here's a very important center of the brain. Let's talk about some memory centers in the brain that we've discovered thus far in science. So one is the amygdala. This is the area of the brain that appears to associate emotional feelings and sensory input with memories. For example, sensory input. I go and I smell a certain perfume and it takes me back to my grandma, right? Or there's a song that's out. I don't even remember the name of the song, but it talks about, I wish I had a candle that, had the smell of my childhood, because I would light it and then you could remember everything that related to your childhood. That's your amygdala. That's actually making that association with emotional feelings and sensory input. But then we've got the hippocampus. And this is the area of the brain that is important in the storage and the retrieval of memories and the transfer of short-term memories. So the hippocampus is a very important part of the brain. And one thing that I want to point out, when you see where these regions of the brain that are very vital for memories and associations, they're in a protective area of the brain. They're just not on the outside where they could, easily be damaged. They're actually, deep inside the brain to protect them from damage. So I do want to make you aware of, when we're talking about the brain and neurons, of a word that you will hear quite a bit called reuptake inhibitors when you're talking about drugs. So drugs and medications such as cocaine and Prozac, these are reuptake inhibitors, and they can cause a prolonged action of a specific neurotransmitter in the synapse. So for example, cocaine inhibits the reuptake of dopamine, which is why they get this cocaine high, right? The dopamine is staying in the synapse. And Prozac inhibits the reuptake of serotonin. So serotonin is going to remain longer in the synapse of the neurons. So that's what reuptake inhibitors mean. And so here we're just, taking a closer look at what that looks like on the molecular level. So here we've got a blood vessel. We're looking at the blood-brain barrier. Here in white, we can see the drug molecules that are in the bloodstream because they were taken in some way by pill, nose, whatever. And then we've got the neurons in the brain that we can see here. And in step one, after the drug is taken, it crosses the blood-brain barrier, and not all drugs can cross the blood-brain barrier, but some can. And if they do, they're quickly, they reach the brain's pleasure centers, these very specific drugs, right? When we're talking about dopamine and serotonin. Of course, there are others. We saw acetylcholine. So they depending on their function. So here we're speaking specifically about dopamine and serotonin. So these drug molecules then bind to sites on the presynaptic membrane, where neurotransmitters are normally reabsorbed by the cells that originally released them. Remember, when the neuron releases the neurotransmitter, they can reabsorb it afterwards, right? Sort of getting ready to fire again if it needs to. Well, these drugs and medications can actually block those uptake centers, right? Those uptake regions of the neuron. And as long as reuptake sites are blocked by the drug molecule, then the neurotransmitters previously released by that neuron will remain in the synaptic cleft. And then these neurotransmitters are repeatedly stimulating the postsynaptic cell, and you can cause this nonstop activity. And if it is in the pleasure center of the brain, you get this nonstop activity in the pleasure center of the brain, and that's why people get addicted to cocaine, right? Or you get what's called adrenaline junkies. They're constantly wanting to do something that is causing a release of dopamine, stimulating the pleasure center of the brain. And hopefully, they're doing something in a safe manner that would cause this release. But we do, as humans, tend to do things that will cause the release of dopamine in the pleasure center of the brain. But let's talk about the thing that we all use that basically has made Starbucks so popular: caffeine in coffee, right? The majority of us, not all, but the majority. How does caffeine work? So here we've got adenosine, which is what we're looking at as our neurotransmitter. So we can have adenosine generated from metabolic activity. And so adenosine, this cellular waste product that builds up in the synapse, binds to the receptors on nearby cells. So here we can see the receptor binding of adenosine. And as more adenosine receptors become filled with adenosine, the neurons in our brain become less likely to fire. And we perceive this as tiredness. And so basically, caffeine is actually interceding in this tiredness action potential, right? And so ingesting caffeine circulates to the brain, binds to the receptors intended for adenosine, and then the adenosine receptors bound to caffeine do not reduce our neurons' ability to fire, and we feel more alert. And that's basically how caffeine is keeping us awake. It's binding into the adenosine receptors. But, when we look at alcohol, so alcohol, let's look at some of the effects. Alcohol can slow reaction time, slur speech by blocking receptors for glutamate, provide a pleasant buzz by blocking dopamine reuptake, and block pain by stimulating the release of endorphins. This, is not a good thing. You want to feel pain, as we know. It increases feeling of happiness by modifying, increasing the efficacy and the efficiency of our serotonin receptors. So it's actually modifying quite a few of our neurotransmitters, right? But this can lead to some very reckless and dangerous behavior, right? All right, so that's going to conclude our talk on neuro, eye, ear, brain, and muscle. Thanks for watching.